So, am I audible to everyone?
Yes, you are audible.
Okay. So, I hope I'm clearly audible to everybody. So, today, hi, I'm Ananya. I'm Ananya Chatterjee. I'm currently a PhD student in ISC Bangalore. So, I'll be helping you with this courses, experimental biotechnology this semester. So, uh, So I am the teaching assistant or TA of this course. I hope the you have already gone through the lectures which are getting uploaded in the SWAM portal, and along with the lectures, If you have any doubt regarding any assignments or regarding any regarding the course or any questions, you can join this teaching assistant lecture series, which will be happening on every Tuesday from 6 to 8 p.m. I'll be taking this session. This is mostly a doubt clearing session, and also I'll be covering whatever the professor is taking. So those sorry, those parts I will also be covering. If you have faced any problem in the lecture or in understanding, you can just get back to me. You can type your questions in the chat section, and I'll be happy to discuss about that. If I'm not so sure, I'll be get back to you with the answers in the next class. So keep it a bit interactive. We will be discussing. If any questions, if I am also not sure, I'll be get back to you with the answers and with the clarifications. And like that, this lecture series will be going on. And I just wanted to know from which background and which standards are you from, like BSc or MSc, like BTech, MTech. uh, few can write in the chat section, then I will have an idea. Mm, yes, someone has raised hand. You can type your question or you can speak out also. But I think there will be some problem in echo. To better if you just okay, MSc. If you have any question, please type in the comment section. BSc third year Biotech. Okay. MSc. Good evening. BSc, MSc, MTech, BTech. Okay. BHMS, BTech Biotech. BE Biotech, MSc Biochemistry, BSc Biomedical Sciences. MSc Biotech. Okay. BSc Zoology. Gayatri, if you have any question, you can just type it. Oh, sorry. I think, yeah. So, okay. So, everyone, okay, one person is PhD. Okay, maximums, all of you are BTech or MTech or MSc BSc. So, you have, I think, gone through the courses syllabus. So, I'll be sharing my screen. Window, entire screen. Okay. So hope my screen is also visible to you. Now, so I have already introduced myself. Yeah. So I have already introduced myself. I'm a third year PhD student in ISC Bangalore, and today is the first class of first tutorial class of this experimental biotechnology course in NPTEL, which will be the duration of this course is July to October 2023. And I think the exam, some of you and all of you will be registered for the courses and will be appearing for the examinations. So beforehand, I would like to inform you is that inform you all that you have to, okay, so you have to submit all the assignments due time. if you are willing to get registered for the course, because some of like 30% or some of the percentage of these final examinations will be from the assignments. So it's better if you just go through the lectures. If you have any doubt, just get back to us, like teaching assistants, to get your doubt solved, and then submit all your assignments timely, and prepare well for the final exams. So, today's class is more of introductory class. So from next week I'll be starting from basically week 1 course. So this is week 0, or you can say that introduction of those lectures what you have seen. But the another important point is that whatever assignments you are getting a due date, right? So I'll be helping you in understanding the concepts, but before due date I won't be able to discuss those assignments which are due, right? So today I'll be discussing with you the mock assignments which was there. I think you have gone through, but I won't be able to discuss week 1 assignments, which is uh, like still online, like you can still will be able to answer and then submit. So I won't be able to go through that. I'll be going through that when your assignments due date will be over. So next week or next week to next week I'll be going through. And another thing, for getting an idea what kind of questions and what kind of things are getting asked or getting discussed in the courses, you can also go through previous lectures of Professor Vishal Trivedi on these courses in YouTube, NPTEL lectures, and whatever assignments were given, you can solve, and I will also be discussing those previous years assignments with you in every week, and we will be discussing those question answers and whatever professor is teaching during the lecture series. Okay. And in this tutorial sections, from today what is I am teaching, this tutorial section I am going to upload in my YouTube channel, which is NPTEL courses. I will give you the link, and also the link will be provided from NPTEL code team also to you, because those lectures will be archived, so that you can just go through later and see. So more of the topics will be, so the first week 0 contents are some introduction to basic lab safety, lab protocols, how to use different instruments and all. Those will be like to get you accustomed with the lab thing. So this experimental biotechnology will be like a course which will be helpful in all techniques aspects. So for basic people, whoever is there, you guys will be preparing for masters, eventually for PhDs, and so for that, for BSc people or MSc people who are going to further for PhD or other entrance exams, and for PhD also, for 3 sections, BSc, MSc, PhD, like bachelor's, master's, PhD, every section, this course is very much important, as you will going through as we'll go through and see, because all the techniques we use during our research all will be covered in this course, and you need to understand while doing those experiments, you need to understand the concepts behind it. Okay. While why you are doing electrophoresis, what are the mechanisms. So just be careful in understanding, be careful in like discussing doubts if you have any doubts, and also so it's a very important aspects with respect to, like from buffer making to different assays, like immunoassays, ELISAs, and also we'll be discussing, and it will be interesting. I think you will like throughout the courses. So at first you have, I think you have seen one mock assignments is there in the portal. If you have not gone through yet, please go back and go through. I think some of you will be like some of the questions you will be able to answer. Some of the questions you may face some problems. Get back to me next class. I will be going through, and today also I have prepared the slides with the answers, but in from the next class I'll be, if you are interested, I'll be just asking you guys answers, and then I'll be discussing the answers with you with the clarifications. So the some of the topics I have not started covering, and also some of the topics are not yet being taught or already taught in the courses, but it's just the mock assignments to get you understand or make you familiar that what kind of questions or what kind of topics you are going to get to know. Okay, so the first question is that which of the below mentioned reagent is not a chromogenic HRP based reagent? So what is chromogenic HRP based reagent? You have to know this. Okay, so this all of these things will be discussed very detailed manner, just to give you an idea what kind of questions or what kind of topics are being taught. So now some of you might heard about Western blot, some of you have not. So for that, if anyone has any question, please you can speak up. Okay, so chromogenic HRP based substrate. I think some of you have heard about Western blot. So what is Western blot? We know protein, right? I'll be going through from very basic to very advanced also. So now I'm going through from basic. So we know protein, and during our experiment sometimes we need to isolate proteins, and we need to confirm whether we have successfully isolated our proteins. So for doing that, we know that we will run a SDS page gel. So to get the proteins, different proteins separated according to their molecular weight. So during that we get to know that we have got a band. I'll just do this. So this is the SDS page MIT. So we run SDS page, so we get to see this bands of protein. So suppose my this is a band of around 70 kDa. Okay, around 70 kDa, and our protein of interest is also of 70 kDa. But in this world there is not a single protein of 70 kDa molecular weight. We need to know what protein is that. So how to know that? Any idea? Can some of you just type in the comment section if you have idea of Western blot or in this situation. So we have to for this experimental design or for different like interviews and all, you will be asked this kind of analytical questions, very basic experimental questions you have to analyze, and you have to come up with some ideas. So in this situation, do you know that what should we do? Like if there are 4 proteins, every protein has molecular weight of 70 kDa, but we need to be sure that this is X protein, this is Y, this is Z. Yeah. Audio is not clear. Is it with everyone? Audio is not clear. Devangana has said. Okay, I think Devangana, you have some network issues. Okay, so people are answering good, very good. Identification by hybridization after Western blotting, fluorescent level, perfect. markers, ladders, yeah, ladders we use to get to know that which size. And after that we'll be doing western blot, or we have to hybridize with some other substrate which are fluorescently tagged. Okay, so I'll be discussing what is that. I'll be discussing what is that. So now what do you do? These proteins we incubate with some other substrates. Okay, suppose that is primary antibody. Now what is primary antibody? I am explaining western blot. Okay, what is primary antibody? Another protein which is the antibody against your protein of interest. So in this case your protein of interest is antigen. So we know that antigen antibody concept in this course also will be discussing, but the basic thing we know that antibody binds with antigen. Okay, so in this case a protein will be recognized by its primary antibody. So it will be coming and it will be binding. After that this primary antibody can be fluorescent tagged and will be able to see under like fluorescence sensor. But if the primary antibody is not fluorescent tagged, what we can do, we can again add another antibody which is secondary antibody which has affinity towards primary antibody. Okay, and what happens in general for chemiluminations, HRP based western blot, what happens, this secondary antibody will be HRP tagged. Now what is HRP? HRP, the full form is horse redox peroxidase. So it has property, it has some property. What it will do after it has so the secondary antibody will have HRP tagged. So what we'll do, we'll add some substrates which are substrates of this HRP enzyme. Okay, this is enzyme. So after adding those substrates, HRP will react and some color will come up. So that is why, because of this color coming up, it's called chromogenic. Okay, so this reaction is called chromogenic HRP based reagent. Anybody has question? Gayatri and Hari Narayan has raised their hands. If you have any question, you can comment also, or you can speak up also. So this is chromogenic, which is colorimetric HRP based reagent. Okay. Yeah. So the concept is that from what you have not understand, Sayan? From the beginning. Okay, I'm just explaining from the beginning. So the concept is that you have your protein in your gel. So during western blot, what do? You transfer the protein onto a membrane. Okay, I will explain the western blot things in more detailed manner when we'll be in that stage. So just for brief understanding to solve this question is that we'll be transferring. Okay, now proteins are on the membrane. How do you know that your protein is there? So the membrane, you will incubate the membrane with primary antibody. So primary antibody is something which has affinity, primary antibody against your protein of interest. Okay, this is the main thing. So this primary antibody will go and bind with your protein only onto the membrane. Then you will add some secondary antibody. So secondary antibody will be against your primary antibody. So then secondary antibody will go and bind to this primary antibody. So here is your protein, primary antibody is coming and binding, secondary antibody is coming and binding with the primary antibody, and secondary antibody is HRP tagged. Now in that membrane you are adding some substrate which are HRP substrate. So HRP is an enzyme which acts on this surface, okay, and generate a color. So this is a principle. and then if you see that color, you know that there are that reaction happened, you know that the reaction happened, and then if only one of particular positions those reactions will be happening, that means there only secondary antibodies are present. And if secondary antibodies are binding there, that means strongly binding there, that means there are primary antibodies which are strongly binding there. Okay, so that means your protein is there. So now the question is that which of the below mentioned reagent is not a chromogenic HRP based reagent? So some examples you need to know what are chromogenic HRP based reagent. So as I was discussing, that which remain in solution, those chromogenic HRP substrates remain in solution and become colored after reaction with HRP. HRP is tagged with secondary antibody. So this can be you can be able to use with ELISA and other colorimetric assays. So some commonly example of commonly used chromogenic HRP substrates are TMB, full form is 3,3' 5,5' tetramethyl benzidine, 2,2' azino di 3-ethyl benz thiazolide 6-sulfonic acid, ABTS. TMB, ABTS are common. There are other chromogenic HRP based reagents which are DAB tablets. So DAB tablets are composed of colorimetric precipitating substrate that produces a brown color upon reaction with HRP and can be dissolved in water to prepare a ready to use buffer solution for blotting detection. So DAB tablets you can be able to use. So TMB is often used as a substrate for HRP in ELISAs. However, in the presence of HRP and peroxide, a water soluble blue product is generated that can be precipitated onto a membrane. Another chromogenic HRP based reagent is 4-chloronaphthol. It is used for both blotting and immunohistochemical staining, but it is less sensitive than other precipitating HRP substrate. And for lab thing, if you are working with that substrate, you need to know you have to store it at minus 15 degree or minus 20 degree and protect from light. Luminol. Now luminol is an exception. It is not a chromophore because if you use that, light won't be generated after HRP catalyzed reaction. This is the thing. Luminol we can use for detection of protein, but in that case light is not generated after HRP catalyzed reaction. So the answer will be, which is not a chromogenic HRP based reaction, the answer will be luminol and H2O2. Okay. Okay, you didn't understand why we are adding secondary antibodies. Okay, if your primary antibody has not any fluorescent tagged molecule, okay, if it is, then you can be able to just see in any detector. But if you don't, your primary antibody doesn't have any Alexaflor or something tagged or any tag, then how you will be able to know that your primary antibody is there? So for that, another molecule we are adding, which is a protein, which is a secondary antibody getting attached with the primary antibody, and which that secondary antibody has HRP. Okay, so this is doubly, we are confirming doubly that primary antibody has bound, then secondary antibody has bound, which has HRP. If we are adding substrate for HRP and getting color, that means secondary antibody has bound. Yes, it's only because of generating color. Yeah. So now in the mock assignments, the next question was, in SDS page, the proteins are separated based on their, so options are charge, PI of the protein, 3D structure, molecular weight. So I hope some of you will be able to answer. So I think you have already seen the answer, molecular weight, but why not charge? Can you please tell very briefly in comment section? In HDS page, why we, like, what is the differentiation factor that protein is getting separated based on what? Is it charge? Yeah, correct. So Khushi has given answer that due to action of SDS. So now I'll be explaining briefly, because we will be going through this very detailed, like in one lecture and all, this SDS page, Western blots and all. So just to answer this question, I'm briefly explaining what happens. So from here only I'm explaining. So here you have run your protein, okay. Now what is SDS page? What SDS do? Your protein is, at first your protein is like that, is forming a tertiary structure, but SDS, what this do, we add SDS in the loading buffer also, in the gel also. SDS linearize the protein and gives a all over the protein a negative charge, okay. Whether the protein is positive charge, negative charge, whatever, it gives the protein negative charge. And after that we use some denaturing substrate also, which like denatures the protein, and also we use like BME, DTT, which reduces disulfide bond. So protein already like lost its all secondary structures, and it just the peptides, like bonds are intact, and just a chains of like primary structure, you can say. So in this situation, the proteins secondary and tertiary structures like are not there, because everything has denatured and like bonds have been broken, disulfide bonds and non-covalent fractions. So in this case, we can't separate every like proteins, because all are now negatively charged, can't be separated based on their charge, can't be separated based on their pi. In SDS page we can, in 2D SDS page we can, will be discussing, but in 1D SDS page we won't be able to, because the pH of the SDS page buffer will be same. In the resolving and the stacking will be different, but in the one resolving in all over the gel, the pH will be same, okay. So we won't be able to detect by pi. Also there is no 3D structure remained, so we won't be able to differentiate by 3D structure. Only thing is molecular weight. Whether like if your protein is of 300 amino acid, the proteins will be of 300 amino acids only after like getting it linear or whatever. If the protein is of 20 amino acids, the peptide will be of 20 amino acids only. So the differentiation factor for SDS page will be molecular weight, okay. Now the next question is, the native page cannot be used to determine amino acid composition of proteins, study protein-protein interaction, monitor in-gel catalytic activity, determine the 3D structure of proteins. So what is native page? Native page is there when you do not use SDS. So here is SDS is denaturing and is making every protein, every thing at like negatively charged, but you don't do those things in native page. So in native page, your proteins are not getting denatured. If your proteins are of oligomeric or multimeric, it will be of multimeric or oligomeric. So the native page cannot be used for what? Options are: determine amino acid composition of proteins, study protein-protein interaction, monitor in-gel catalytic activity of enzyme, determine 3D structure of protein. So the 3D structure of the protein we cannot tell through a native page. The 3D structure, we can't get to know what is the structure of the protein. We can get to know that how much molecular weight of is this, like, so the study, the, yeah. So, and also if two proteins are interacting, we can be able to tell that these are interacting. So the answer is that. Now the next question is that the purpose of glycerol in HDS page loading dye is to. So while we load our protein in gel, in the loading buffer, one substrate we also add some amount of glycerol. So why do we add that? Options are increase the density of sample to facilitate loading, prevent degradation of proteins, reduce the clumping of different proteins present in sample, provides uniform negative charge on proteins. So the answer will be the increase the density. You know that glycerol are a bit viscous and it adds density to the sample so that the sample goes into the lens. So the gel runs like this, right? Vertically, so goes into the lens and go. Then prevent degradation of the proteins, not correct, and provides the uniform negative charge, it's function of SDS. Now the next question is, the main purpose of the layering of dissolving gel solution with organic solvent during casting SDS page is to. Now we'll be discussing this in detail. Again, I'm telling you that. So while preparing the gel, if some of you have not done, we won't be able to get it until we discuss in detail. Still, whoever has done or whoever has gone through those lectures. So while we cast our dissolving gel, no, we add little bit of isopropanol onto the layer. So why do we do that? Options are reduce the evaporation of dissolving gel, which is not correct, prevent rapid solidification of gel, which is not correct, prevent entry of oxygen into dissolving gel, absorb extra moisture from gel. So the answer is prevent entry of oxygen into dissolving gel so that random oxidation doesn't happen. So this gel buffers contains APS, SDS and all, so which can get oxidized. APS can get oxidized. So to prevent entry of oxygen from the air onto the dissolving gel, so we add organic solvent little bit, and then when the dissolving gel gets solidified, we just remove it. So this is the principle. Now the next question is, in native page, the protein's migration is based on what? So what happens in native page? We do not add SDS, we do not hit the protein so that the protein is not denatured. Now here SDS is not there, so all the proteins will not be in peptide chains and will not be in like negatively charged linear. It's not like that. Proteins will be of same molecular weight, how much ever it has, and whatever like oligomeric or something like that, it will be like that only. So now how we'll be able to differentiate between different proteins, and migration will be based on what? So migration will be based on the charge of the intrinsic charge of the protein and also the mass. So charge to mass ratio at running pH, the pH will be using. So at that pH, whatever the protein's amino acids, like whatever charge it retains, that charge by mass ratio will be determining factor for protein migration. Okay. Now the next question is, a protein X has a molecular weight of 80 kDa, okay, and it is a heterodimer. How many bands you will observe on gel if you resolve the protein on SDS page and native page? Okay. Now, to make you understand, if you are running a protein which is oligomeric, here it has written that which is heterodimeric. What is heterodimeric? Means a protein is made up of two different proteins, A and B. So heterodimer means both A and B doesn't have same molecular weight. They do not have same molecular weight. It cannot be 40-40. Okay, those will be of different molecular weight and different like subunits. So how many bands you will be observed in SDS page? So we in SDS page we know that we denature every like secondary structures. So we'll be getting only one band. We'll be getting so it will be denatured. So we'll be getting for both the subunits. When we are getting like for the heterodimer we are denaturing it, so we'll be getting two bands. But for native page we'll be able to see the exact molecular weight it has, the whole oligomer. So we'll be getting one band. Okay. So while we'll treat SDS and we'll boil it, the heterodimer it won't be like remain as heterodimer, it will be monomeric. So we'll be able to see only two bands. Now the tracking dye used in SDS gels is bromophenol blue, the tracking dye. So while we make loading buffer, we also load some dye to track that proteins are moving. The dye from the dye front we see. So in that what dye we use? We use bromophenol blue. Okay. Now the purpose of SDS is to provide, I have already discussed, which is answers like options are a negative charge to protein which are positive charge, a uniform positive charge to proteins, uniform negative charge to proteins, positive charge to the negatively charged proteins. So it's not like that it gives negative charge to positively charged protein. It's not like that. It's not like that positive charge to negative charged protein. No, it gives a uniform negative charge to all proteins. Now the another question is, urea page is used to study the folding states of protein, 3D structure determination, glycosylation status of the protein, amino acid sequence of the protein. So urea and guanidine hydrochloride, it's used for denaturing protein and like denaturing, and then if you remove the urea it will be helping in refolding. So to determine or to study the folding states of protein, we use urea page, and in details I'll be covering in the next class, in electrophoresis class. So the solvent used to charge the PVDF membrane during Western blotting is methanol, to activate PVDF membrane so that it becomes hydrophilic and proteins gets transferred. Okay. So this all the like topics are not yet covered, I know. We'll be going through all the topic in very detailed manner. We'll be covering electrophoresis, agarose gel electrophoresis, SDS page electrophoresis, Western blot, and whatever I have discussed to the native SDS in detailed, and how to make protein loading dye, and what are the constituents we'll be discussing. So today is a mode of introductory class. Now for the introduction of these courses, I think you have gone through some guidelines and precautions. So this course will help you in like getting you into the laboratory also. So you need to, before starting working in a laboratory, you need to know some basic precautions, guidelines for your for doing the experiments. So let us see what all the things are important for laboratory course, especially a course utilizing biological specimens, chemicals, possibly harmful radiation, and some do's and don'ts we need to know for wet lab course. Okay. So I'll be just going through these do's. which are always wear the appropriate equipment of personal protection. They include an apron, pair of gloves, safety glasses, shoes. Read thoroughly the detailed procedures before carrying out the experiment. Identify the potentially hazardous materials required for performing the experiments. Okay, it's recommended to go through material safety data sheet supplied with the chemicals to understand their potential hazards and safe uses. Before starting the experiment, label all your materials, tubes, vials, whatever. Check the label on the container before taking out any substance. Use a clean spatula for taking out solid chemicals. Be well aware of the locations and operating procedures of the safety equipment such as safety shower, first aid kit, fire extinguisher, eye wash. Okay. Always use a chemical fume hood for strong acids and other fuming chemicals. Use the recommended biosafety level containment while handling microorganisms. Before using a biosafety cabinet, always ensure that the UV lamp is turned off. Okay. Decontaminate your workbench and the equipment handles after use. Decontaminate all the infectious materials before disposing them off. Maintain a laboratory book and write down all the methods, procedure, observation in detail. Any spillage, accident, or injury should immediately report it to your lab instructor. Now, what are don'ts? Do not sit on the laboratory benches. This can be contaminated with some strong chemicals and all. Do not sit there. Never eat or drink anything inside laboratory. Do not handle your contact lenses or apply cosmetics in the lab. Never touch any chemical or reagent with bare hands. Never try to test or smell any chemical reagent. Do not pipette any of the chemicals through your mouth. If our chemicals are taken from original container and then you happen to take out more than the amount you require, never transfer back the excess amount to the original container. If concentrated acids are to be diluted, never add water to the concentrated acid. Never. Concentrated acid should be added to the water drop-wise with continuous stirring. Never leave your experiments unattended. Never point the open end of any glassware or vial towards yourself or any other person. So these are some signs you can see in the laboratory. So these are some symbols of inflammable, oxidizing, acute toxicity, and corrosive. Okay. Now some biosafety levels or biohazards you can see. So low risk microbes are number one, which are of low risk, which are not known to cause disease in healthy adult humans. Minimum potential hazard to laboratory personnel and the environment, example E. coli. So while we use E. coli for bacterial experiments, it's of risk, but it's very low risk. Second or moderate potential hazard to laboratory people are different aerosols, which are hepatitis A, B, C, influenza A, Lyme disease. Level 3, microbes can cause serious or potentially lethal disease through respiratory transmission. When if you are working with some viruses like HIV, flu, which is influenza H1N1, influenza, tuberculosis, SARS, rabies, West Nile virus, you have to be very careful because these are of serious lethal disease. Now, high-risk microbes are dangerous and exotic, posing high risk of aerosol-transmitted infections. Infections caused by these microbes are fatal and without treatment of vaccine. There is no treatment, so Ebola, etc. So you have to be very careful while doing your experiments in the lab. Okay. So some guidelines and precautions you have already gone through in the lectures. So which are recommended action? Decontaminate all the infectious material before disposing them off. Sanitization, disinfection and sterilization. What is sanitization? Surfaces clean. Do clean your surface, microbial load reduced. The common solution for routine cleaning. A mild cleaning agent such as soap or household detergent will clean greasy surfaces but may have little antimicrobial activity. Disinfection. Microbes on surface killed, inactivated, and made harmless. The solution for simple cases for contamination: use quaternary ammonium compounds which have antimicrobial property. They do not corrode metal or damage plastics. Sterilization: total removal of any contamination, no viable microbes remain. Reserved for serious contaminations. Use oxidative agents such as bleaches, hydrogen peroxides. Stick to the safety rules and do not use these agents on metal or plastics. Okay. Any spillage and accident, do report immediately to the lab advisor. Now, I have already gone through this do's and don'ts you need to know before doing experiments. Now, some instruments. So pH meter is one of the important instruments you will be using for making your buffers, for setting pH of those buffers. So what is pH meter? So this is kind of plastic and some glass material you can see in your lab to measure your pH. So what are the different parts of this? So it has a reference fill hole which is used to replace the reference electrolyte solution. Okay, there is two electrodes or elements which are pH internal elements which supplies a voltage based on the pH value of the sample. So this pH meter you dip into the solution and it gives you a value. So there is a digital monitor attached with the pH meter, so you will get a value. And there is another internal reference which supplies a constant equilibrium voltage. Okay, and there is a sensing membrane glass which performs the actual function. So pH meter or pH measurement requires two electrodes. One is pH dependent electrode which is sensitive to H plus in your solution or in your buffer, and another is pH independent reference electrode. So how pH meter measure pH? So they measure the potential difference between these two electrodes. Okay, the potential difference between two electrodes is measured as voltage, which is given by V equals to electrode E constant plus 2.303 RT by FP. So this is the formula. So here V is the voltage, E constant is the potential or reference electrode, potential of reference electrode, R is the gas constant, T temperature, and Faraday constants, from where it calculates like the difference between the voltage, it calculates and gives the value. Now the courses introduction we have already gone through that whatever the topics we'll be covering. So at first, good laboratory practice, we have already gone through those good laboratory practices, and we'll be going through introduction to water and buffer, because water or molecule or distilled water will be the one thing which will be using regularly while doing the lab work, from making buffers to running gel to running any experiments, protein purification, everything you will be needing this molecule water. And module 2 will be covering so yes, so this module 2 is it's not in the this year topic. I will update you in the next class again. So this is I have taken from the previous year's lecture course syllabus. So the next topic, so this topic won't be covered in this year. So the next topic will be electrophoretic experiments for separating and characterizing the proteins and nucleic acids, which I will discuss in the next class. Then module 4 will be chromatographic experiments for separating, purifying, characterizing biomolecules. Module 5 will be different amino acids like ELISAs, immunofluorescence. Module 6 will be methods for studying cellular structure, components and all. Module 7 will be discussing the basics tools and experiments of molecular biology. Okay, we'll go eventually into that stage. Now, as a like introductory class of today will be discussing basic proteins and nucleic acids and what are the constituents of those. So chemical and structural features of proteins and nucleic acids. Okay. So we know that proteins constitute the cellular machinery, okay, that carry out majority of the biological reactions. You will be get to know that this protein is important for doing these things. This protein is is toxic to these cells and like that. So, so proteins are also made up of amino acids. You know that. So they are linear polymers of L alpha amino acids. The structure of a typical L alpha amino acid is shown in figure. You will be see. So this is a basic alpha amino acids. So this is alpha carbon. You can see that one carboxyl group is attached to the carbon, one hydrogen, one amine group, and another group which is R, which can be hydrogen or can be some bulky groups. So this is the basic structure of amino acid. Then the R group, also known as side chain of an amino acids, and makes a repertoire of 20 different amino acids. So if R groups gets changed into different chemical structures, we will get 20 different amino acids. So in proteins, amino acids are linked together through amide bonds formed between alpha amino group and the alpha carboxylate group. So you can see the amine bond in between alpha carbonyl group and alpha amino group and alpha carboxyl group. So that is this bond. Okay. So when two amino acids get linked by peptide bond, one molecule of H2O gets released and the bond is made up of one carboxyl and one amine. Okay, C double bond O, single bond NH. So this bond is peptide bond, which happens in between two amino acids. Now, based on the chemistry of the side chains, amino acids have been classified as polar and non-polar. Polar amino acids have been further classified into neutral, acidic, basic amino acids depending on the charge on the side chain at neutral pH. Example, lysine side chain, for example, has a terminal amino group that is protonated at neutral pH. Therefore, it's identified as basic amino acids. Aspartic acid and glutamic acid have carboxyl group in their side chains. At neutral pH, the side chains are ionized, making them acidic amino acids. Okay. So the amino acids in a protein are linked through amide bond, which is also called peptide bond. Delocalization of the nitrogen's unshared pair of the electron over carbonyl group impairs a partial double bond character. So this is C double bond O, this is NH. So delocalization of the nitrogen's unshared electrons over carbonyl group impairs a partial double bond character. So this bond attains a partial double bond character, and this implies that the rotation about the C double bond O NH is not allowed. Okay, putting constraints on the conformations of the polypeptide backbone. The rotation about C alpha and CO and N and C alpha, however, are allowed and are thus called dihedral angles, referred to as psi and phi respectively. The amino acid sequence of a protein, which is also termed as primary structure, is read from N terminal to C terminal. The polypeptide region within a protein can adapt local ordered structure very similar psi and phi values for a continuous stretch of amino acids called secondary structure. So we have heard about alpha helices and beta acids. Okay. So these are two important secondary structures. Further assembly of the polypeptide chains leads to a compact structures called the tertiary structure. So this is primary structure, stretch of amino acids, then alpha helix, beta sheets. This is one tertiary structure which is composed of different secondary structures, and this is quaternary structure which is composed of different tertiary structures. Okay. Now, another important thing to start our laboratory experiments to know nucleotides and nucleic acids. So, a nucleotide is a monomeric unit of nucleic acids. Okay. Chemically, a nucleotide is composed of a pentose sugar. You can see a pentose sugar or ribose sugar. Okay. Attached to the heterocyclic nitrogenous base, which is heterocyclic nitrogenous base, can be purine, can be pyrimidine, through N-glycosidic linkage. Here, this bond is N-glycosidic linkage, and to one or more phosphate groups. One or more phosphate groups. Here, 3 phosphate groups are attached. Okay, through phosphoester bonds. The pentose sugar can be of ribose if the nucleotide is called ribonucleotide, and can be of deoxyribose. So deoxyribose, when this oxygen won't be there, then it will be deoxyribose. Okay. So this is 3. Okay, so 2 prime oxygen won't be there, so this is 3 prime oxygen, so it's not this. I'll be removing. Okay. So is called deoxyribonucleotide. So nucleic acids are also polymers. Okay, so nucleic acids are polymers of nucleotides. Nucleotides, the polymer of nucleotides is called ribonucleic acid or RNA, while the deoxyribonucleotides is called DNA. Now, so this is the nucleotide. You can see that different nitrogen bases, pyrimidine, thymine, cytosine, uracil, and this is ribose sugar. This is deoxyribose sugar, where 2 prime oxygen is not there, and these are purine. So in pyrimidine we have thymine, cytosine, uracil, and in purine we have adenine, guanine, and this is a tRNA molecule. So just one example of RNA, and this is double helical structure of DNA, one example of DNA. Okay. So the bases present in DNA are adenine, thymine, cytosine, guanine. In RNA, the thymine gets replaced by uracil. We know that DNA is usually made up of 2 antiparallel chains coiled around each other in a double helical structure, okay, or double stranded DNA. Adenine, thymine, adenine, uracil, guanine, cytosine are said to constitute the complementary base pair. So adenine makes double hydrogen bond with thymine, G triple bond with cytosine. The 2 strands of DNA molecules are antiparallel and complementary to each other, that is, thymines and cytosines on one strand are paired with adenine and guanines respectively on the other one. RNA molecules are typically single-stranded but can be of double-stranded as well. RNA molecules can have intramolecular complementary sequences, causing them fold into defined 3-dimensional structures, example transfer RNAs. Nucleotides such as ATP, GTP, flavin adenine dinucleotide FAD, nicotinamide adenine dinucleotide NADP+, coenzymes play critical role in metabolisms and intracellular signaling. Now we have got a basic idea of protein and nucleic acids. Now we will discuss a little bit of water and aqueous solutions. So water is the most abundant chemical component present in living system. We know it constitutes around 60% or more of the total mass of most of the organisms. The first organisms are believed to have originated in the aqueous environment. Most of the biological reactions are carried out in water, many of them directly involving water as one of the reactants or products. It will not be incorrect if we call water as a biological solvent. To understand the chemistry of the biological processes and the behavior of biological molecules in water, we need to understand the physical and chemical properties of water. Biochemical experiments are mostly carried out in aqueous solutions having a defined chemical composition and pH. So in this today we'll be discussing the physical and chemical properties of water, concepts related to solutions and pH. So hydrogen bonding and physical properties of water you already know. So This is water molecule. This is the structure of water and hydrogen bonding. So this shows a bent structure of water molecule. So the big red sphere represents the oxygen, while small spheres represents the hydrogen atoms. Uh, and in panel B, those H2O are making like bonding each other with hydrogen bonds, and shows the hydrogen bonding in between water molecules. Okay. So water is a molecule wherein oxygen atom is sp3 hybridized. Two of this hybridized orbital make sigma bonds with hydrogen atoms, while the other two are occupied by the unshared pairs of electrons. Water therefore is a bent molecule, okay, with an H angle of around 105 degree. The large difference in the electronegativities of oxygen and hydrogen make OH a very high polar bond, because oxygen has negative potential and is positive. Okay. The large difference in the electronegativities of oxygen is making highly polar bond. Owing in the angular geometry of the molecule, the dipole moments of the two OH bonds do not cancel each other out, impairing a high dipole moment. So water has a high dipole moment of 1.85 dB. The oxygen atom is partially negatively charged, while the hydrogen atoms are partially positively charged. This leads to an electrostatic attraction between oxygen atoms of one water molecule and the hydrogen atoms of one another water molecule. The interaction is referred to as hydrogen bond. So it is due to this hydrogen bonds that water molecules are held together, making it a liquid at room temperature. So because of the hydrogen bonds only, all water molecules are linked together and making hydrogen bonds and making it liquid. So it is due to like hydrogen bonds between these water molecules impart it high melting point, okay, boiling point and heat of vaporization. Low molecular weight and the ability to form hydrogen bonds also impart water a very high specific heat. It takes 1 calorie to increase the temperature of 1 gram of water by 1 degree at standard temperature and pressure. Water therefore is an excellent temperature buffer. Hydrogen bonds are not restricted to water. They are readily formed between any electronegative atoms called a hydrogen acceptor, usually fluorine, nitrogen, oxygen, and then hydrogen attached to another electronegative atom called a hydrogen donor. Hydrogen bonds are weak but play a very crucial role in the structure and function of the biomolecules. Common hydrogen bonds are observed in biological system will be showing. So in panel B shows like hydrogen bonding between water molecules. Panel C shows the hydrogen bond is frequently formed in different biomolecules. So polar solutes can form hydrogen bonds with the polar water molecules, thereby dissolving in water. So if one polar solute we just drop onto the water, so water is also polar, the solute is also polar, so like dissolves like, so polar solute like substrate will be dissolved in water. Non-polar solutes on the other hand do not make favorable interaction with the water molecules and clump together. Water can easily dissolve charged solutes by hydrating and stabilizing the solute ions. Sodium chloride, for example, readily dissolves in water. The Na plus and Cl minus ions acquire greater freedom after leaving the salt crystal lattice. This increase in entropy makes the overall free energy change negatively, therefore favoring dissolution. The compounds that readily dissolve in water are termed as hydrophilic, while those do not dissolve are termed as hydrophobic. So this is some chemical properties of water. We know that water is weakly ionizable, and it gets ionized into H plus and O plus, H plus and OH minus. So this is how the equation goes, that free H plus do not exist in water and they exist at hydrated ions. So H plus again associated with another H2O, it makes H3O plus, which is also called hydronium ion. And the dissociation of water molecule can be expressed in the terms of equilibrium constant. So equilibrium constant equals to H plus into OH minus divided by H2. Okay, this is equilibrium constant. Dissociation of water is so weak that the concentration of undissociated water can be assumed to be constant, 55.5 molar. So also, so 55.5 molar to K equivalent equals to H plus into OH minus equals to Kw. You can also write it. So where Kw is ionic product of water. Okay. So now will be, so you can see that the concentration of H plus and OH minus at 25 degrees Celsius for pure water is 10 to the power minus 7 molar each. Therefore, the ionic product of water is 10 to the power minus 14. When the concentration of H plus and OH minus are same, the water or any aqueous solution is said to be at neutral pH, that is pH 7. So this is the pH scale. So if you take logarithm of the previous equation and you multiply by negative charge minus log pH, you know that this is pH, minus log H plus, this is pH. So pH plus pOH equals to 14. So like that, the above discussed definition of pH does not pose any limitations on the values it can take. It's however convenient to define a pH scales from like 1 molar H plus to 1 molar OH minus. The pH scale thus obtained ranges from 0 to 14. It should be clear by now that pH is a scale for defining the acidity and basicity of aqueous solution. A solution having a pH less than 7 is acidic. So whenever you are making a buffer and the pH has become like 4 5, it's acidic. And whenever the buffer like pH is, whatever solution pH is greater than 7, it's basic. So pH is an important concept for living organisms. Now expressing concentration. So while we different like solutions and different preparation we do, the calculation we do based on some units which are molarity, normality, like that. So what is molarity? So as most biological processes are carried out in the aqueous environment, it is important to know how the concentrations of the solutions are expressed. So concentrations based on volume. Concentrations represented as the amount of substance per unit volume of solution are the most widely used in a biochemical laboratory. So what is molarity? Molarity is the number of moles of solute per liter of solutions. 1 mole is defined as the amount of substance that contains as many atoms or molecules or other elementary units as the number of atoms present in 12 gram of carbon 12. So 1 mole is a mass in grams numerically equal to the formula weight of the substance. So number of moles equals to mass of the substance in gram divided by molecular weight of the substance. Normality, how to measure the normality, is the number of equivalents of solute per liter of solution. One equivalent of an acid or base is the mass that contains 1 mole of repressible hydrogen or hydroxy. One equivalent of a compound involved in oxidation reduction reaction is a mass that donates or accepts 1 Faraday of electrons. Okay, this is how we calculate equivalence. We had those topics in 11th 12th, so we are just revising this. So concentration based on weight. So we also measure concentration like weight by volume, like that. So what is weight by weight percent? The weight of the solute in gram per 100 grams of solution. Molality, the number of moles of solute per 1000 grams of solvent. Mole fraction of a compound in a solution is a fraction of the total number of moles represented by the compound in question. Like if a solution contains N1 moles of compound 1 and N2 moles of compound 2, N3 moles of compound 3, then the mole fraction will be of solution 3. Suppose mole fraction of solution 3, that is N3 divided by total mole fraction, that is N1 plus N2 plus N3 plus N4. Okay. Now again coming to the buffer. So what do buffer do? Buffer maintains the pH of the solution. So we shall be discussing about the ionization of weak acids and preparation of buffers using weak acids and bases. So you will be now, so after going through the lectures, if you also do practical courses, you will be able to make and prepare buffers to any strength, any concentration and any pH. So ionization of weak acids and weak bases in biochemistry, it's convenient to use Bronsted-Lowry method or concept of acid and base. So a substance that donates proton is acid, while a substance that accepts proton is base. When an acid loses a base is produced. So and the acid and the corresponding base are called are like said to constitute or conjugate acid base pair. Okay. So here HA plus B minus equivalent to A minus plus HB. What is happening? HA is releasing one H plus and becoming A minus, and B minus is taking up H plus and becoming HB. Okay. So we'll be discussing these equations later words. It's just a complicated. Uh, you have all gone through in 11-12 standards. So just how to deduce the Henderson-Hasselbalch equation. If you like just go through the equation, you will be able to understand that if you add water in the HA, it will become H3O plus or hydronium ion. Then the ionization can be represented as equilibrium constant. So equilibrium constant will be this into this divided by this and this into this, and we know that the concentration of the undissociated water we can consider it as 55.5. So if it goes into this, then H2O into K equivalent, that is 55.5 molar into K equivalent equals to this. And by solving this and by taking log and then taking negative charge, you will be getting one equation which is pH equals to pKa plus log A minus HA. So where P is a shorthand for negative logarithm, and this equation is called Henderson-Hasselbalch equation. So pK can be defined as a pH at which acid is 50% ionized. So we do titration of a weak acid. So what happens? Example of weak acid is acetic acid. So when titration of a weak acid with a strong base, that is sodium hydroxide. So what happens? Addition of sodium hydroxide. If we add sodium hydroxide in acetic acid solution, so addition of sodium hydroxide neutralizes the H plus ions present in the solution, thereby increasing the pH. Okay. Consumption of H plus ions drives the dissociation of Ce3. So whenever the H plus ions will be getting degraded, disrupted, so what happens? CH3COH can be dissociated into CH3CO minus and H plus, right? So according to La Chatelier's principle, whenever the H plus is getting dissociated, this rate, the dissociation rate of CH3COH will be increased. As more and more of NaOH is added, the pH of the solution increases. This increase in pH is small when the solution reaches the pH close to the pK of acetic acid. A weak acid therefore provides a resistance to change in pH near its pK value. This resistance to change in pH by an acid near its pK value is called the concept of buffer. Okay. So this is a titration curve. OH minus added, pH value is getting changed, but near the pK value, the pH is not getting affected that much. This is the buffering region. Now some basic concepts of centrifuge. So in the laboratory, we'll be end up using centrifuge to precipitate out different solutes. So this is suppose different solutes are dispersed in a solvent, and after centrifugation you can see this is a clear solution where all the solutes are getting precipitated. So what happens in centrifuge? Centrifuge happens separation through sedimentation could be done naturally with the earth cavity. If you keep for very long time one solute which is not suspended or completely dissolved in the solvent, then you can eventually see that the solutes are getting sedimented. okay, because of the gravity. So centrifugation is also like that, but it's making the natural process much faster. So while what we do in centrifugation, we rotate our substance, and rotation of the rotor about a central axis generates a centrifugal force. So one centrifugal force is getting generated, and there a centripetal force opposite towards, okay, and which helps in the precipitation of the substance. So rotation of the rotor about the central axis generates a centrifugal force. You can see F1. F1 is a centrifugal force upon the particle in the suspension. Which factors have an influence on centrifugation? So what are the factors that have influence on centrifugation? Density of both the sample and solution. So whatever sample or solution you have kept, what are the density of that? Temperature and viscosity of that. Okay, what is the temperature of the like solution? What is the viscosity? Is it too viscous? It is less viscous? Distance of particles displacement and rotation speed. So here you can see this, here centrifugal force is acting there, frictional force will be acting opposite side, it will not help the centrifuge, and the buoyancy will be act also opposite side. So sedimentation will be happening if F1 force, centrifugal force, is greater than F2 plus F3, okay. And in density gradient, what happens? F1 equals to F2 plus F3, because what happens in density gradient, solutes are getting separated according to the different density. So in the density gradient, the solvents have different density in a gradient wise, so those will be getting sedimented in their same density. So at a fixed centrifugal force and liquid viscosity, the sedimentation rate of a particle sedimentation rate of a particle is proportional to its size and to the difference between particle density and the density of the solution. So in centrifuge also, you have to remember, you have to balance the samples. So whatever centrifuge which goes to 5000 G, we can balance by pouring equal volume liquid in other centrifuge tubes. Centrifugation beyond 12,000 G, you should balance the sample with weighing balance within range of milligram. Centrifugation ultra speed, that is ultra centrifugation, you have to balance, should be very accurate. If you use centrifuge at low temperature, after centrifugation there will be condensation of water, okay. Wipe the centrifuge cup with dry cloth. You need to keep the lid open so that all water should evaporate. Now for your experiment, preparation of water, why there is need to purify the water. So ordinary tap water contains a variety of impurities including particulate matter, sand, silt, etc., dissolved organics, inorganics, and gases and microorganisms. In addition, it may also contain metabolic byproducts from microorganisms called pyrogens. Distillation, ion exchange, carbon adsorption, reverse osmosis, membrane filtration, these are done for water purification. Water purified by deionization, reverse osmosis, distillation usually is acceptable. Water that is purified by ion exchange may contain certain organics that are washed from the ion exchange regime. These contaminants will increase the ultraviolet absorbance measurement are to be made. Distilled water is better than deionized. Okay. So solution, a liquid mixture in which the minor component, that is the solute or the powder, is uniformly distributed within the major component, that is called liquid mixture. So solution is made up of powder plus solvent or liquid plus solvent. Okay. So we have already gone through the concepts. Again, the most common units used for preparing the solutions are molarity, normality, percent by weight, percent by volume, weight per volume. Molarity, which is concentration based on the number of moles of solute per liter of solutions. It can be millimolar, micromolar, nanomolar, like that. Molecular weight of glucose is 180. We know this volume is 100 ml. So to prepare 100 ml of 1 molar glucose, what you have to do? You have to add 18 gram of glucose, okay? And then you will transfer to a 1 liter volumetric flask, add about 700-800 ml of purified water, swirl to dissolve, then add water so that the bottom of the meniscus is at the H line. Stopper and mix well. The flux must be labeled. So how much molar? 0.1 molar glucose, date prepared, and the name of the prepared. So this is how one perfect like buffer or solution you need to make like that. Okay. Normality is a number of equivalents of solutes per liter of solution. Percentage per weight by weight, concentration based on the number of grams of solute per 100 gram of solution. Percent by volume, concentration based on the number of grams of solute per 100 ml of solution. Weight per volume, concentration based on the number of grams, milligrams or micrograms of solute per unit volume. Okay. Now pH we have discussed this concept. So this is a pH scale. Here you can see below 7 this is a lower pH acidic substance, and above pH those are like alkaline substrate. Okay. Now buffers also we have explained that buffer solutions are used as a means of keeping pH at a nearly constant value in a wide variety of chemical applications. In nature, there are many systems that use buffering for pH regulation. Example, bicarbonate buffering system, which is like carbonic acid H2CO3, bicarbonate ion HCO3 minus carbon dioxide maintain the pH of blood to support proper metabolic activity. So blood pH we know around 7.4. So why there is need of buffer? For example, the role of pH in controlling biological processes. So enzymes are proteinaceous in nature and made up of individual amino acids with ionizable side chains we know, such as histidines. In addition, active site of enzymes has also amino acid residue, and a particular ionization state is important for substrate binding, formation of catalytic intermediates and release of products. For example, pepsin is a serine protease present in stomach and has an optimum pH of 1.5, where trypsin has a pH optimum of 7.4. Okay, so we need to have some buffering agent so that pH is not getting like higher and lower. It should be maintained. In many pathological conditions such as diabetes, body utilizes stored fat as an alternate energy source. The similar condition exists in the case of starvation or fasting, and under these conditions a large amount of acid beta hydroxybutyric acid from fat is generated, leading to lowering blood pH called acidosis. It disturbs activity of several enzymes present in blood, ultimately leads to headache, nausea and convulsions. Blood pH is maintained by bicarbonate buffer system and play a vital role in respiration. So optimum pH is needed for every like activity of different enzymes. So here are some enzymes important for our human body, that is lipase, pepsin, trypsin, urease, invertase, maltase, amylase and so on. And you have seen that how buffer maintains pH. So buffer is a mixture of weak acid and its conjugate base, or vice versa. So in the above ionization reaction, what is happening? Ha and A minus make one conjugate acid base pair, while B minus and Hb make another conjugate acid base pair. So when Ha dissociates, it becomes H plus plus A minus. Now if you add strong acid to it, what happens? Ha plus H plus will be H plus plus Ha. Now if you add strong base, it will become H2O plus A minus. So buffering capacity is a quantitative measure of the resistance to change of pH of a solution containing a buffering agent with respect to a change of acid and alkali concentration. Okay. Now we have gone through the titration of weak acid with weak base already. We have also gone through Henderson-Hasselbalch equation. We have seen preparation of buffer. Now, preparation of buffer example we have seen. Again, if you are given a point that you have to prepare 500 ml of 200 millimolar phosphate buffer at pH 7.4. So you need to know what are the constituents of phosphate buffer. You need to know the molecular weight, okay, to make a phosphate buffer, because you need to calculate the molarity. Now, like that, you will be making those buffer. So they have, like in the lectures, two methods have been discussed. So what you will do, you will weigh your uh, whatever constituents are there, you will weigh accordingly. 200 millimolar, you need to calculate that how much gram you need to add to make it 200 millimolar. So we know from the molecular weight, we know that this much gram is for 1 molar, so how much will be for 200 millimolar. You will add that, you will add distilled water or milky water, and then you will after dissolving, you will like reach the certain volume which is required, and you will measure your pH. If pH is not exact, you have to add HCl, you have to, or you have to add NaOH to decrease or increase your pH. Okay. So this is how we will make so this is the calculation. You have to calculate molarity, and then you will add, like, check your pH, and then you have to make up your volume. Okay. So this is all for today's class. If you have any doubt, you can let me know. We'll be discussing in the next class. uh, Regarding assignments also, if you have any doubt, you can tell me. I can help you out, and we'll be discussing in the next class regarding electrophoresis, SDS, gel electrophoresis, Western blots. If you want to if you want some topics to be discussed, you can also let me know. We'll be discussing those topics in the next class for your understanding. Okay, what did you say about spectroscopy? Is it in syllabus or removed? So, spectroscopy in this syllabus, in the current ongoing courses, spectroscopy is not there till I know, as far as I know. But in the next class, I will be telling you, because your syllabus will contain whatever the lectures are going on. So the first topic, you have I think gone through those basic lab protocols, how to make buffers and all this. The second lectures will be electrophoresis and all, and then the third lectures will be for techniques, another technique. So spectroscopy was there previously when the course was being taught by two professors, but now the course is taught by one professor only. So now the spectroscopy has been removed, as far as I know. Any other question? What did you say about spectroscopy? Okay, can you please share the details on YouTube? Yeah, the details of YouTube channel I'll be sharing. I'll be sharing in the next class after I upload this video. So the next class I'll be sharing, and I'll be like yeah, I'll be sharing in the next class, and also it will be shared by NPTEL team. This today, so every day's we like lectures I am recording, and I'll be uploading in the YouTube. You can see anytime. Can we have this presentation? Yes, the presentation I will also the presentation also I will share with NPTEL core team. I won't be able to share you directly. I'll be sharing with NPTEL core team. They will be there is a like data sheet where they will be uploading those lecture recordings and videos, presentations, everything you can get from there. Good. Can you explain pK portion, how to get those values? pK portion, okay, so how to pK, so every pK if you go and like go through some biochemistry like books, you will have those pK values. Okay, the pK values are known, and you can also like calculate those pK's values, how I was showing, but those values are like known for those amino acids, 20 amino acids. Can you register for 2 exams going to happen in same day? You can register, but if you are like able to manage to be appear on the 2 exams, like if one is in first half, another one is second half, you can apply, you can register. What is the method used to studying protein interaction, type of page used to study protein in case of diabetes or fasting is reduced, which causes acidosis in stomach? These are 4 options in assignment: hydrochloric acid, beta butyric acid. How can we find answer when we are not able to find in lecture slides and material? Please suggest. Okay. Okay, so the mock assignment, the question was that what is the study, and this lecture is not being taught. So that's why I'm telling you, if like some lecture slides are not there, like, so what you have to do, the old lectures are still available in YouTube. Okay, if you are finding any problem in one assignment, I will be telling you the answer and the I will explain, but I can't explain before submitting the assignment date. So you just go through the NPTEL. The professor's lectures will be available in NPTEL Experimental Biology. Biotechnology. If you type this biotechnology, if you type this, you will be able to see the professor's video, previous year's video. You can just go through those topics, and you can get to know that which acids are like causing those acidosis, and for what reason. So you'll be able to understand. And the next class, if you have any doubt, after the due date, I'll be explaining you. Ma'am, would this course help us to design experiment? Yeah, it will be helpful. It will be, because you will be getting a sense of what is the principle behind those experiments. And in the professor's video also, in the last sections of those classes, you will be having some experimental problems and analyzing, and I will also if I am also able to find some experimental questions, I will also put in my lecture. So it will be helpful to design experiments, some sort of experiments. You need to solve some problems, you will be able to design, because behind like designing the experiment, you need to understand what principle you need to what principle you need to come up with. So you'll be able to kindly share the YouTube link too. Yeah, YouTube link I'll be sharing. So currently, actually, the previous year also I taught some NPTEL one NPTEL course. So that YouTube channel only I'll be uploading these videos. So I'll be naming the YouTube channel today, and the link I'll be sharing in the next class, because this video I'll be sharing, and then only I'll be sending you the link. Deva and Ghosh, why does the SDS page and or native page run vertically and DNA gel electrophoresis run horizontally? Okay, so the principle between horizontally and vertically, there is no big difference. So what happens, the agarose gel we make through agarose, so it's not like it's getting oxidized and making or reduced, making the gel is polymerizing. It's not like that. It's a solid gel, and we don't worry about getting oxidized while we make the gel, so we make it solid and horizontally. And for SDS page and native page, we use APS. So we to like prevent oxidizing, we need to have like the gel, the polymerizing casting between two glass plates. So it's better to have like in vertically. And there is the same principle behind is that it the the gel runs accordingly, not accordingly gravity or something. It runs accordingly electrophoresis, like voltage difference. So there is no principle. Isoelectric focusing, yeah, isoelectric focusing also we'll be discussing. Whole proteins, yeah, yes. This is all, I think. How frequent will be these live sessions? Okay, so this live sessions will be each week, one day. Tuesday every week, Tuesday every week till your exam comes up, like when the course ends, uh, 6 to 8 p.m. Also, every classes live sessions video will be recorded and will be uploaded in a YouTube channel. You'll be able to go through. Any question, anyone? Okay, so if you don't have any questions for today, you can please join the next class next Tuesday, 6 to 8 a.m. 8 p.m. Sorry, evening 6 to 8 p.m. We'll be starting electrophoresis principle, from basic principle to some advanced principle, some problems, and we'll be also discussing some assignments. So thank you so much for attending the courses. Thank you all.
Thank you, ma'am.
Welcome. Bye.